Welcome back, everyone, to the Diaspora Dialogue. I'm Sudeep.
And I'm Bhaskar.
As we briefly mentioned in our first episode, our very own Bhaskar is a microfluidics expert. He did a PhD and years of post PhD work on it. We are going to dive deeper into this technology. So, I have personally been excited about this episode for a long time. This excitement is because we are talking about a technology that most people have probably never heard of, but nearly all of us have interacted with it almost every single day.
Exactly, Sudeep. Today we are talking about microfluidics, which, in very simple terms, is science and engineering of controlling extremely small amounts of liquid inside microscopic domains. Honestly, when people first hear the word, it sounds very abstract and overly technical. But once we start seeing examples, it suddenly becomes very intuitive. For example, trees use natural microfluidics to transport water and minerals from soil through roots to all the way to leaves. Blood flows through microscopic capillaries in our body. Even our lungs rely on very precise fluid and gas transport. Also, the paper towels we use every day utilize capillary force to soak water.
Bhaskar, you are defining this microfluidics as such a simple phenomenon. Why is this simple phenomenon interesting for applied researchers like us?
Let's think about it this way. Let's ask a simple question. What happens when we shrink the fluid systems down to extremely tiny scale? And surprisingly, when you shrink that to small scale, the entire behavior of fluid flow changes and the principle behind them changes as well. That's what makes this field so fascinating.
Man, now you are going to make it like a dense engineering lecture. Please, listener, don't worry. We'll make it very conversational. What I love about this topic is that it sits at the intersection of biology, chemistry, physics, medicine, and engineering. And even though most people don't realize it, microfluidics is already behind pregnancy test, rapid COVID test, glucose strips, DNA sequencing platforms, drug discovery systems, and even some advanced food technologies.
Yes, Sudeep, we use microfluidics in the background of all of those technologies you mentioned. And in many ways, it's one of those invisible technologies quietly shaping the future of our healthcare system. Most people never directly notice microfluidics itself. They notice the final result or the experience after that result. They notice that the tests become faster, diagnostics become portable, and a system becomes much cheaper. Then finally, the biology becomes more automated. But underneath all these improvements, there's often some form of microfluidic engineering helping make it possible.
Also, Bhaskar, this is a topic both of us have some connection to, right?
Definitely.
I have seen microfluidic system during research environment and academic settings. But Bhaskar, you have spent years and years directly working in this field, from graduate research all the way to the projects at the Wyss Institute at Harvard. And honestly, one reason I was excited to do this episode is because I have personally seen how naturally Bhaskar connects engineering with biology.
Thank you, Sudeep. What attracted me to the field originally was that microfluidics never felt limited to one discipline. As a student coming from an engineering background, I was fascinated by the idea that something as simple as controlling tiny droplets or tiny volumes could suddenly become useful for medicine, diagnostics, biology, food systems, and even material sciences. During my master's work, I started getting exposure to microscale systems and learned how fluid behavior completely changes at the small scales. During my PhD, I became much more involved with droplet microfluidics, biomedical systems, and translational applications. One thing I really enjoyed was that the field consistently forced people from different backgrounds to collaborate. You would see engineers talking to biologists, chemists talking to physicists, clinicians talking to material scientists, and so on. And later, at the Wyss Institute, that interdisciplinary environment became even more intense.
That's something I have heard consistently from people working in advanced biotech. I feel like the boundaries between fields start disappearing.
Exactly, Sudeep. And the microfluidics is one of those technologies that sits right in the middle of all these different disciplines. So today we'll mix the history of the field, the science behind the microfluidics, the practical applications where the technology is heading towards, and some real world stories from the research and translational environment.
And hopefully, by the end of this episode, our listeners will start noticing microfluidics everywhere.
I agree, because once you understand it, you start seeing it all around you.
That's true, Oscar, and that's the game of our brain. Once you are introduced to an object that you have never seen before, then you start seeing it everywhere. For example, Oscar, have you been to Pokhara?
Yeah.
Have you seen an image of a tiger in Machu Picchu,
Yes.
the Fistel Mountain?
Yes, someone showed me when I was there.
That is seen
Yeah.
from Pokhara side, right?
Yeah.
Once you see it, you can't unsee it.
I know. Yeah, every time I see the picture, I always realize there's a tiger.
Right. Now I think we are derailed enough. Let's come back to our topic, microfluidics.
Okay.
So, Bhaskar, let's start simple. What exactly is microfluidics in terms of engineering? Now we talked about plant pulling water from the
Soil.
grounds up to the leaves and paper towel soaking the water. In engineering term, what is it?
At the core, microfluidics is the science of controlling very tiny amounts of liquid inside microscopic domains. It can be channels, droplets, or so on. The easiest way to imagine it is, what if we could shrink the entire biology or the chemistry lab into a tiny chip? That's basically the dream of microfluidics.
Wow. And what people hear tiny, they probably still imagine something visible.
Definitely, Sudeep. I used to do the same thing before I started microfluidics. But in microfluidics, tiny becomes extremely tiny, such as 1 millionth of a meter is a micrometer. That's where the name comes from. For comparison, one single strand of our hair is less than 100 micrometer. So we are working in that scale. Sometimes researchers work with droplets smaller than what a human eye can clearly see. And what becomes fascinating is that fluids stop behaving the way we expect. At a normal scale, fluids are chaotic, coffee swirls, rivers become turbulent, but when we bring that to the microscopic scale, fluids become smooth and predictable.
So I have heard the term laminar flow. Is this where laminar flow comes in, and what is it? Could
Yes.
you please explain it a little bit?
Yes, Sudeep, that's where the laminar flow comes in. As I mentioned, chaotic flow. That's means like there's like all mixing around, and there are like eddy currents, like the circular currents, all those things in the chaotic mixing. And when we bring that scale into micro scale, what happens is the fluid starts moving in layer by layer. That means layer on top of other layer, and the interaction between those layers are in such a small scale that becomes irrelevant. And now suddenly the flow becomes more predictive. That predictability allows scientists to control chemistry and biology with incredible precision. And honestly, that realization is what made the field explode scientifically from last few decades.
So you are saying we have seen these techniques being used in nature, for example, to transport water by the water and minerals from soils to leaves. Blood flows through microscopic capillaries in our body and our lungs as well, right? So
Yeah.
in many ways, we are basically scientists are learning from the biology and recreating this principle artificially and using them for applied systems, right?
Yeah.
One thing I find interesting is that microfluidics feels like a field created by multiple scientists, revolutions happening at the same time, right?
Yeah. I think there are two things. One, as you just mentioned, we brought these phenomena from nature, and all the scientific fields, they came together, and the revolution happened combining them together. So what happened is, analytical chemistry wanted faster and smaller systems. Genomics required massive automation, millions of parallel processing or parallel experimentations. Microelectronics inspired people to miniaturize those devices, and the biodefense program started funding these portable detection technologies, and all these worlds started overlapping. That's where microfluidics started.
And that's probably why the field feels so interdisciplinary. Bhaskar, as we mentioned in our introduction, we'll talk about some history behind it and applicability of it. Before we dive into the applicability, let's talk about tiny bit of history about this microfluidic system and technology.
Yeah, naturally, engineers, scientists, chemists, physicists, biologists, everyone started collaborating together. With this collaboration, what happened is this big lab scale that becomes miniaturized, and then they started putting the name for as lab on a chip, which started being popular these days. The idea here was very simple. Can we shrink workflows in the lab into tiny automated systems?
So you are basically saying previously, probably you have seen like a previous very first round of computer pictures. Like there used to be a room full of computers and machines in there. Nowadays, almost all of us, have tiny computers in our pocket as smartphones, right?
Yeah. Sudeep, I don't know if you have seen the Mark 1 or Mark 2. That was one of the very first computer built in the US. That is in the engineering building at Harvard. I don't know if you have seen that.
Oh, I have not.
That's like a whole room full of computers, like one single computer. That's fascinating. And as you said, that big scale computer came into like our palm and in the pocket.
Right.
Exactly, that's what happened with the microfluidic systems.
So let's make this practical. Where do normal people encounter microfluidics? As we mentioned trees and paper towel and everything, but technology-wise, using engineering this, learning from this nature and engineering this and using in our lives, could you please explain some of the use case scenario for this technology?
Definitely. One of the best example I can give or I can start with is most of our listeners might have seen the home pregnancy test. That's one of the example where microfluidics is used as a practical application.
Engineered and then utilized for a practical application.
Definitely. And what's fascinating is that almost nobody looks at a small plastic stripe and thinks about advanced engineering behind it. But internally, it's a beautifully designed fluidic system. And the user experience feels extremely simple. You just place a drop of sample on the stripe, wait a few minutes, and there is a result for you. But underneath that simplicity, the device is carefully controlling fluid transport, reaction timing, biochemical interactions, and signal generations. And the amazing part is it often works passive. There's no batteries, no pumps, and no electronics.
So for this pregnancy test as well, you are saying the liquid moves primarily through capillary action.
Yes.
This is more or less the same phenomenon that allows water to travel through paper towels when it touches water, or plants pull water upward through the tiny vascular channels, as you mentioned earlier, up to the leaves, right?
Yeah, exactly, Sudeep. And one more thing to mention here. Inside that pregnancy test, there's a paper stripe in between that plastic. Now there's the paper flow that you mentioned about the paper towel. That's exactly the same phenomena there.
Right. And as you mentioned, biochemical reactions there has been introduced into the paper.
Yes.
That you mentioned, and that reacts with the solvent that provided in the liquid that is provided into the test.
Yes. The reagent in the stripe is already built in. It's dried out and built in the paper.
Right.
Now, when the sample from the sample from the pregnant women that goes through the paper, it flows through the paper and goes to the point where that dried reagent is. That's where the signal comes out.
Right.
Regarding the applicability, even something people consider it very ordinary, ordinary consumer products is actually built with an elegant microfluidic principle.
Right.
Just we mentioned.
As you mentioned, the pregnancy test is an example. Another huge example recently is COVID rapid test.
Absolutely.
Which we all of us did, right?
Yeah. I think one of the good things of this pandemic is the pandemic was made the microfluidics or made the public exposure to the microfluidic technologies. Millions of people suddenly started using rapid diagnostic systems at home. And what looked like a simple disposable stripe was actually the result of decades of workflow in fluid control, material science, assay development, and manufacturing engineering on that scale. It's interesting because many people experience advanced diagnostics becoming decentralized for the very first time in this.
Right. So the blood sugar testing strip is also an example of it in the healthcare examples,
Yeah,
right?
that's
How
very
about
commonly
beyond
used.
in healthcare? Do ball pen and fountain pen that we used to use in past, or sometimes we use it now, use the same technology? And what else?
Yeah, definitely. That's a great example because I don't think this generation, the new generation, know about this fountain pen.
Right.
We grew using fountain pens.
Right.
Yeah. In between the metal and the plastic part, in the
Mhm.
tip of the pen,
Mhm.
there's a small channel which is a microfluidic channel,
Right.
and that certainly uses capillary force.
And
And
the printers used to be the microfluidic system in our age, right?
definitely, those inkjet printers
Mhm.
are classic microfluidic examples. What they do is they generate precisely controlled mic droplets and then they spray on paper.
Mhm.
And that's how the printing is done.
That's true. I never thought about printers that way.
Yeah, and again, DNA sequencing, they use DNA, like a lot of microfluidics in sequencing these days. For example, the other examples can be drug discovery systems, environmental sensors, industrial dispensing systems. Even it's not very commonly imaginable that airflow sensors use microfluidics as well.
So this field quietly spread everywhere, right?
Exactly, it's like an invisible infrastructure behind all those technologies.
Right. So Bhaskar, why does microfluidics become visually fascinating? What's the physics behind it? As an engineer, if you can explain that in layman term, that would be great.
Definitely. One of the reasons microfluidics become visually fascinating is the physics starts feeling different
Mm-hmm.
than what we generally see. At the microscopic scale, surface tension and viscosity, they both become dominant.
Mm-hmm.
A droplet in that scale becomes soft, like a soft dynamic object
Mhm.
or a gel droplet. And researchers now can manipulate these fluids or droplets using pressure,
Mm-hmm.
electric fields,
Mm-hmm.
magnetic particles,
Mhm.
or even sound waves.
The sound wave part is still feels slightly unbelievable, Bhaskar.
Yeah, it sounds like science fiction. But yeah, researchers literally use these sound waves, sometimes called surface acoustic waves, to move microscopic droplets using carefully engineered acoustic vibrations. And I also did some work in manipulating these droplets using surface acoustic waves.
Mhm.
Again, surface acoustic wave means the acoustic or sound waves that goes through the surface of a body.
Mm-hmm. And that looks
And
almost alive, right?
Yeah,
I
and
think
those droplets,
that's one of the reasons people emotionally connect to this field. It's scientifically beautiful, Bhaskar.
yeah, the droplets, they look like a cells coming alive.
Right.
And sometimes watching these laminar flow
Mhm.
droplets under the microscope, genuinely we see like microscopic choreography.
Mm-hmm. So one of the most exciting areas in the field is probably droplet microfluidics.
Definitely,
That
yeah.
has been used in my field. I'm from biology, so I have seen droplet PCR, which uses single droplet to sequence to amplify single RNA molecules. That's fascinating, Oscar.
Yeah, definitely. The idea is very surprisingly simple.
Mm-hmm.
What it is each droplet can be a reaction chamber.
Right.
Now, when you have million cells, you distribute them like individual droplets.
Right.
Now you look them under the microscope and do whatever you want to do with
Right.
the assay systems.
Mhm.
Now imagine generating thousands of millions of those droplets
Mhm.
and running thousands of millions of reactions at the same time.
Mhm.
Now biology becomes suddenly massively scalable.
So you're saying instead of running one experiment at a time, researchers can run enormous number of experiments simultaneously. For example, I have billions of neurons in my brain. If I want to know what is happening inside those billions of neurons, then you can generate billions of microfluidics droplets and incorporate my neurons into those droplets, and those droplets would behave individually. You can run reaction individually and read them individually and understand what is going on with each of my neurons, right?
Exactly, yes. Now, as you mentioned, we can distribute each individual neurons that you have into individual droplets.
Mhm.
That's why microfluidic droplets becomes important for genomics. enzyme screening, synthetic biology, single cell analysis, and so on. And because the droplets are tiny, experiments become faster, cheaper, and very high throughput. That means millions at a time.
Right. So Bhaskar, you also work on your master as well as PhD work. This is where some of your work connects, right?
Yes, I did use microfluidics or a flow system. I used droplet systems as well. In some of the projects I worked, microfluidics was mainly used as a precise droplet generator. The goal was generating highly controlled microparticles and encapsulate biological molecules in them and make uniform properties of each droplets. And that's important point. Sometimes microfluidics is the final product. Sometimes it's an invisible enabling technology behind the other applications.
Right, Bhaskar. As a biologist, I could say microfluidics also became deeply connected to modern biology. As I mentioned earlier, more automated and data-driven researchers needed system capable of handling huge number of experiments efficiently. Microfluidics has become one of the technologies enabling that transition. Now, especially for things like genomics and single cell analysis, Bhaskar, could you add a little bit more from your side as well?
Yeah, like as you mentioned, we have started thinking about like millions of experiments at a time. Now researchers realize that averaging all the signals across million cells often hides important biological behavior. Now as we started distributing them, now we start to get individual signals from individual cells or individual droplets, and that helps scientists to isolate and study individual signals there independently.
Right.
And you can completely change how researchers or how we think about biological variability there.
Right. So coming out of the cells and getting into the organ system now, I'm surely our listeners have heard of organ on chip system, and which has been a common ground for you and me. Could you explain a little bit about this organ on chip system?
Definitely. Let's think about it. We are made of multiple organs. And what if we can bring those organs in a chip form,
right,
that we can use it for different experimentation? So
right,
we don't have to use ourselves. Instead, we can use the cells from our body and test it there. That's what is called organ on a chip. Researchers have started building these microfluidic systems that can recapitulate or mimic some behaviors of some of our organs, like lungs, intestines, liver tissues, and so on. The idea here is to recreate more realistic environment or biological environment instead of those petri dishes we use.
Right.
Sudeep, you used some of the organ on chip at the WISH Institute, right? How was your experience there?
Yeah, basically, before I joined WISH, as I mentioned in our previous episode, I started working with organ on chip system. For example, liver on chip, that was exciting for me. And stomach on chip. Now I'm working on cancer on chip, lymph node on chip, those projects.
Cool, that's cool. And one thing that stood out when I was working, when you are at the WISH, how strongly people get focused on translational impact from these microfluidic systems.
Right.
And the goal with using these microfluidics is just not to build interesting devices, but to answer some of the questions like how can this eventually improve healthcare or overall research.
Right. Now, Bhaskar, let's be a little bit realistic now. So this microfluidics system or technology has been existing for decades, right? As we mentioned earlier, even for years. Why isn't every hospital fully running on lab on chip system yet?
That's honestly one of the most important questions in the field, and I always think about it as well. Because if you read scientific papers from early 2000s, many researchers predicted that microfluidic chips would completely transform the medicine very quickly.
So almost every hospital would eventually become filled with tiny automated chips. Is that what you're saying?
That's what we thought when we were like early 2000, and that's how the researchers thought that time. While the field has made enormous impact, translation into large-scale product turned out to be much harder than many researchers initially expected. Scientifically, microfluidics is incredibly promising, but commercialization has been more difficult.
So what's the bottleneck? What are the limitations?
The major issue is interfacing the microscopic world with the macroscopic world. For example, getting samples in and out of chip reliably is harder than people imagine. Another challenge is manufacturing. Making prototype is something, but it's very different when we start mass producing those reliable cartridges and replicating them. If you ask about the practical bottleneck, personally, I would say air bubbles is one of the hardest thing to work with. Minute air bubble in a microchannel completely shifts the pressure, and the shift in pressure makes it completely unpredictable.
Right.
As you already mentioned, I was working with Microfluidics flow device in one of the projects, and in another one, I was making micro and nano bubbles using Microfluidic devices, and those were for ultrasound imaging and contrast agent or something like that. And those projects were simultaneously running. And on the earlier one, I was trying to avoid bubble, while in the later one, I was trying to make bubbles. So during that time, I had love and hate relation with these micro bubbles in the microfluidic system.
At that time, you may have been viewing the situation through a dualistic mindset.
Yeah.
So the engineering challenge shifts from invention to scalability.
Exactly. There are also regulatory challenges in healthcare. Clinical validations take time, and sometimes existing technologies are already good enough.
So that's something people often underestimate. Our technology does not just need to work. It needs to be cheaper, easier, faster, and more reliable.
Very true, Sudhir.
So where do you think the field is heading next?
I think we are entering to a phase where the science in the microfluidics is getting mature enough that the real world starts to integrate, and we're focusing on this integration right now. We'll probably see more decentralized diagnostics and more automation and more personalized medicine in the recent future.
Right, especially in this world of AI. AI will probably become part of that ecosystem too, right?
Definitely, AI will be there.
Right.
And AI is already there. For example, if we have to analyze thousands of images from these microfluidic chips, we use some sort of artificial intelligence there. And again, as we talked about millions of droplets, there as well, we started, our people have started using AI to analyze those droplets. And also, microfluidic systems, they generate enormous datasets, and AI these days can help them optimize experiment-wise and analyze result much faster.
So with this AI evolving now, I also think portable diagnostic devices and some treatment tools and drug delivery will have huge impact globally.
Absolutely. Affordable point-of-care systems could dramatically improve healthcare accessibility, especially in low-resource settings or developing countries. And honestly, that's one of the most inspiring long-term goals of the field.
That was such a fascinating conversation, Bhaskar. And I think the biggest takeaway is that some of the important technologies are the invisible ones quietly operating behind the modern life.
Exactly, and microfluidics is one of those hidden enabling technologies.
And once you start noticing it, you realize it's everywhere.
Yeah, everywhere, from healthcare to biology to manufacturing to diagnostics.
Thank you everyone for listening to episode 4 of the Diaspora Dialogue. If you enjoyed this episode, share it with friends, family, students, engineer, scientists, or anyone curious about how technology quietly shapes our world.
And if there are topics you'd like us to discuss or explore in the future episodes, please let us know.
Until next time, stay curious, keep learning, and keep the dialogue going.
Thank you, Sudeep.
Thank you, Bhaskar. We'll see you next time.